Hi everybody, this is Dr. Demi. It has been a really long time since I recorded a video and I am delighted to be doing it again. It has been a while. I've been busy with other things, with work, with life, but it is great to always remember that May, June exams are right around the corner. And I saw that I was starting to get questions from people on the channel asking me about certain things. So I put out a post to ask students if there were any things they were struggling with and would like me to do a video on. And I got an overwhelming number of requests for paper 5. So I thought this video would be a good way to address that. So if you haven't been here in a while and you are hoping for some revision for paper 5, please settle in and watch this video. For the most part, I will go through paper 5 from May, June 2023. I'm going through just question 1, but I will also share some tips with you as I go along, which would be very helpful for you as you prepare for your exam. All right, let's get into it. So I'm going to start off with some tips for paper 5 because I find, and when I recall my experience as a teacher, I find that when students encounter paper 5 for the first time, they tend to do really badly at it, even though they do really well in paper 3 or they understand paper 3. So the best way I can describe paper 5 to you is to let you know that paper 5 is about experimental skills, but without the experiment. So it's you doing something without really doing it. So you're thinking about the experiment and you're planning it out, but you're not doing it. It is a planning and analysis paper. It is for 30 marks and it's always 2 questions. The first question is where you do the planning for the most part, and the second question is where you usually do the analysis. If you are able to tackle paper 5 really well, you have a high chance of getting a high grade in biology because it is very easy to get high grades in paper 5. It's just when students don't understand it, then they struggle with it. I will also say that from my experience as an A-level biology student years ago, and then becoming a teacher, I find that sometimes teachers don't go through paper 5 with their students because they assume that the students, having understood paper 3, will be able to do paper 5. It is important to go through paper 5 as well as paper 3, and I'm saying this because I know that there are some teachers who watch this channel, and I'm hoping that this will sort of like serve as a springboard for teachers to understand that paper 5 is equally important to go through. It is a very confusing paper when you encounter it for the first time, so it is important that students are exposed to it frequently. When I was a teacher, my students had paper 5, I think, every week throughout their second year of A-level biology. So what should you know about paper 5? You need to understand that the theory is important, which means that you should understand the principles of the different experiments that you've done in biology so far. So everything you started doing, from enzymes, from everything, just make sure you have learned that properly. And you should also take note of the steps that you would typically do when you're doing an experiment in class, which is why, again, it is important that students are exposed to experiments, because doing those experiments will help you think through your paper 5 properly. The most important thing here is you have to approach the questions in paper 5 as logically as you possibly can. And remember that sometimes the answers are in the question. And I'm pretty sure this question we're about to tackle will show you an example of that. All right, let us get started. The first question for May, June 2023, paper 5, version 1, is about enzymes. So the moment you see catalase is an enzyme, you already know you're doing something on enzymes. Back in the day, I was under the impression that paper 5 only covered the topics that you did in the A-level year, so the last, I believe, 7 chapters of A-level biology, but it turns out that's not the case. You actually have to know and remember everything you've done in AS level these days. So let's go through this question together, and of course, as we go through it, I will give you some tips to help you understand what an examiner would be looking for as they mark your paper. So first things first, the question goes, catalase is an enzyme that is found in many plant cells and animal cells. Catalase breaks down hydrogen peroxide, which is a waste product from cell metabolism. Figure 1.1 shows the breakdown of hydrogen peroxide by Catalase. And now you have this reaction here, which is the first thing you have to take note of, because it says here hydrogen peroxide, when you add Catalase to it, it will yield water and oxygen. So if it's telling you water and oxygen, then you can either already think, just by looking at that reaction, that you would either be asked to estimate the amount of water produced or something along those lines, or to estimate the amount of oxygen produced through bubbles or something along those lines. So just like already getting into the spirit of what could this experiment be about, even though you are not about to do it, you are simply going to plan it. So question 1 is the planning question. Then it goes, a student decided to investigate how catalase activity varies along the length of a carrot root. So it's very clear it's a carrot root. If I was a student doing this paper, I would underline along the length of a carrot root, just so that I am sure of what exactly it is that I'll be working on. And here they've shown you a figure. Don't discard the figures when you get them, because they have very interesting labels, just so that students are not confused. So it says these are the leaves, that's the shoot, which is right at the top of the carrot, and then you've got the root of the carrot, which is basically the entire length here. So it's telling you that it is going through the whole carrot, and not just not the top where the shoot is, and obviously not where the leaves are. So bear that in mind as you start to think of planning the experiment. It then goes, the student extracted catalase from different parts of the carrot tissue. Carrot root tissue was mixed with a small volume of pH 7 buffer and some fine sand. The mixture was ground with a pestle and mortar. The mixture was placed into a centrifuge tube and spun in a variable speed centrifuge for a few minutes. This caused the mixture to be separated into a solid pellet at the bottom of the centrifuge tube and the liquid carrot extract at the top, as shown in figure 1.3, and this here is figure 1.3 on the right. The liquid carrot extract was poured into a clean test tube. This liquid contained catalase. Then it goes, state one variable that should be standardized in the procedure to obtain the liquid carrot extract, other than the carrot tissue, the fine sand, and the apparatus used. Now, again, I want you to put yourself in the position of someone who is doing this experiment in the lab. You've cut the carrot tissue. You've mixed it with a small volume of pH 7 buffer and some fine sand. You've ground it with a pestle and mortar, and then you've placed it into a centrifuge and spun it in the centrifuge for a few minutes. And then it says that it separates blah, blah, blah. What is a variable that you should keep constant here? The first thing that jumped out to me when I was reading just the methods was how long would we keep the carrot mix in the centrifuge for? Because that was not specified. Like, how long should we keep it there for? The other thing that jumped out to me was the volume of the pH 7 buffer. It just says small volume. So I would say perhaps that volume should be constant, because you don't know how it would impact the, I'm just going to write volume of buffer here, how that would impact the experiment. But listen, over here, you only need to supply one thing. This is another thing you have to note. I often find that students, when they write paper 5, because they are so unsure, they want to put in as many answers as possible, hoping that when the examiner is marking it, they would be able to dig through all the mess that they've put down and find the answer. You need to be very clear, which means you have to be confident in your answer when you're doing paper 5. So you only need to put down one of these for you to get 1 mark. Then they go on to tell you what the student did. It says the student carried out these steps to investigate the activity of catalase in different samples of carrot root tissue. A disc of filter paper of 5 mm diameter was dipped into the carrot extract using forceps, and forceps are like tweezers. So if you're someone who tweezes your eyebrows, you probably know what that is. The filter paper disc was then placed at the bottom of a beaker with 1% hydrogen peroxide solution, again using forceps. The time taken for the filter paper disc to rise to the surface of the hydrogen peroxide solution was measured. Figure 1.4 shows the apparatus used, and I will now show you as well on the next slide. So this is figure 1.4, which is this. Again, let's just think back to what the student had already done. The student started off with a carrot. I'm just going to draw a random looking thing here. This is our carrot. It's got leaves on top. And the student took samples along the length of the root because the student was trying to investigate where the activity of catalase might be different. They took those samples and then they ground it rather with sand and a buffer in a tube, which they spun in a centrifuge, and that led to a pellet at the bottom. I'm just going to darken this place to say this is the pellet, and you had a liquid on top, right? Then they took a disc paper and put it in here, and we were told that this liquid on top of the centrifuge tube contained catalase. So that means this filter paper will now have catalase on it. Then we take that filter paper and put it into this beaker here, which has 1% hydrogen peroxide solution. Now, I want you to take note of things on this diagram. Something I've said at the beginning of this video is that when you are doing paper 5, sometimes the answers are in the question. So you have to take note. How much hydrogen peroxide solution did they use? It was 1%. What was the diameter of the filter disc paper? It was 5 millimeters. Some of these might come to use when you get to the part where they ask you to plan your own experiment if you were the student. But that's not the question we've come to just yet. They've just said that the apparatus shows, the figure rather shows the apparatus that was used to measure the time taken for the filter paper disc to rise. State the relationship between the time taken for the filter paper disc to rise to the surface of the hydrogen peroxide solution and the catalase activity of the liquid carrot extract. My first question here is, why would the catalase rise? If you remember, when we looked at the reaction for catalase, or rather, why would the filter paper rise? Sorry. When we looked at the reaction for catalase, it looked something like this. We had hydrogen peroxide that was becoming water and also giving off oxygen. I don't think my equation is balanced. I think that's probably it. Well, hopefully, but let's not go into the chemistry part of it. But anyway, so you know that oxygen is being produced, which is why this paper would rise. And that's an indication that the catalase is working in the paper. So this then says, states the relationship between the time taken for the filter paper disc to rise to the surface of the hydrogen peroxide solution and the catalase activity of the carrot extract. Now, if you think about the effect of enzyme concentration, if you have a high enzyme activity, then you will have high products, like really quickly, right? So the more your enzyme works, or the faster your enzyme works on the substrate, the quicker there will be products. So that means that if we are measuring time here, then we can say that the time taken for the filter paper to rise is a result of the activity of catalase. So you can then go the faster or the higher catalase activity. And please write in better sentences than I am. I'm just showing you what the higher catalase activity. Use your words as well, right? Then the quicker the filter paper rises. Because as catalase activity increases, it will produce oxygen quickly, and that oxygen will sort of buoy the paper up. So the quicker the filter paper rises. And I'm pretty sure there are better ways to put this in the mark scheme, which I think you can look at. But this is just me thinking through the problem with you. And yeah, hopefully nothing is wrong, but I don't think I'll get any of these wrong. I still remember biology quite well, even though I haven't taught it for a while. All right, so I hope that's clear. And don't worry about this here. I just put it here as a reminder of what the previous slide was. Okay, so then the question goes on and it says, the student decided to investigate how catalase activity varies along the length of the carrot root and it just shows you again what the carrot root is. So they're really trying to tell you where this experiment is taking place, and they're saying this entire place here, this entire length, is the carrot root. And you can see they've put a ruler here to say like this, they're measuring the distance from the base of the shoot, which is just right here, right, all the way down. The student made the prediction: as the distance from the base of the shoot increases, the catalase activity of carrot root increases. Identify the independent variable in this investigation. So if you've done paper 3, you've probably seen that sometimes you have to come up with a hypothesis, and they're telling you here that the student has already come up with one. The student has said, I think that as we go down this carrot route, so as we go in the same direction as this arrow on the page, the activity of catalase increases. So that means that there is probably more catalase at the base of the carrot than there is at the top of the carrot root, than there is at the top of it. What would be the independent variable? Always remember that the independent variable is the variable that is not dependent on anything. It just exists on its own. In this case, catalase activity based on the student's prediction would be dependent on the distance from the base of the shoot. So it means that like as you go further, then catalase activity changes, which means catalase activity is a function of distance. But distance is not a function of anything. It's just the length of the carrot, which then means you can go ahead and say that the independent variable here would be the distance. So always remember that the dependent variable is dependent either on the conditions of the experiment or on some other variable within the experiment, whereas the independent variable is not dependent on anything. It just exists. It's just there. Rather, it is the one that maybe plays a role in how the dependent variable rather plays out. So just bear that in mind. All right, now we've come to the favorite question, which is the planning question. This is the question that usually has the most marks allocated to it. And if you get this question right and are able to get maximum marks here, then there is a high chance that you're going to do really well in question number 1. It says here, describe a method that the students could use to test the prediction that as the distance from the base of the shoot increases, the catalase activity of carrot root tissue increases. Your method should be set out in a logical order and be detailed enough to allow another person follow it. Details of how to use the centrifuge to prepare the liquid carrot extract should not be included. So that means they're saying that, you know, that part we told you earlier where we said the student put it in the centrifuge and then spun it and they separated it. That's not important. We already know that part. Tell us how you would plan this experiment. Now, I'm going to do that because I'm going to try to write out as much as possible because I know this is where a lot of students struggle. So this is our carrots that we've been given. Right? We're going to role play. We're going to imagine that you're in a lab coat, you're standing at your desk, and you've been given a carrot and a lot of other tools. And obviously we know what this experiment is. They've given us an idea, which is why I say again that when you're doing paper 5, the answers are already in the question. So first things first, whenever you are taking samples in paper 5 or even in paper 3, always remember that you're going to take a minimum of 5 samples. Okay, always remember that, because you might get to a point where, for example, in paper 3, they tell you to draw a graph. So you should know that minimum has to be 5, and minimum is 5. So first things first, you will start out by saying, okay, I am going to prepare my carrot. Now, you don't need to write in first person to say, I will first do this, I will first do that. It just says describe a method. So you can write it as if you're preparing a method for someone else who is a student to follow. So first things first, you can say cut a long carrot root. And I don't think I'm using the right English here. Maybe I should not say cut along. Should rather say cut out carrot tissue sample at specified lengths of the carrot root. So let's assume, I'm just looking for my pen here, okay? So let's assume you can say this is one length, this is another length, that's 2, that's 3, that's 4, and then this is 5, okay? So you can say cut out carrot tissue, okay, at specified lengths of the carrot. I don't write fast with this pen, but it's the best I have, so we'll just have to work with it. A specified length. All right, so that's one. Cut out carrot tissue at specified lengths, and then you can write here you should cut out a minimum of 5 different samples. Okay, so 5 different samples. Write in fuller sentences than I am, because that would also be helpful. And I'm pretty sure that even just writing these two in this way would probably give you 2 marks. So cut out carrot tissue at specified lengths, and maybe mention a tool that you would use to cut it out. So maybe you can say using the small knife provided on the desk, cut out carrot tissue at specified positions along the length of the carrot root. You should collect a total of 5 samples. Then you want to say the next bit. You don't need to include the centrifuging part, right? Like you can just say for the next bit, you will now weigh, and this is important, weigh 0.1 gram of carrot tissue to extract catalase, for catalase extraction. Okay, I'm just going to write extract like that. Then number 3, you can say prepare 5 centrifuge tubes. Prepare 5 centrifuge tubes, add the carrot tissue for the 5 different samples, and spin. So even though they've said don't include that, right? They've said how to use the centrifuge to prepare the liquid extract. Don't include that. You just need to say that you will centrifuge it, but you don't necessarily need to say, oh, and then I'll spin it at this speed for this long. That's what they're saying you shouldn't include. But you can say prepare 5 centrifuge tubes, and then spin and collect extract. So you might be able to say that, but you can also leave that out, to be honest, because they've basically said the details of how to use the centrifuge to prepare the liquid extract should not be included. But you can say prepare 5 centrifuge tubes and collect what's it called, catalase extract. Then the next thing you would do is you would measure, I can't remember what the volume was of our hydrogen peroxide, but I can just say measure 10 cubic centimeters of hydrogen peroxide into a beaker, and you can even specify the concentration as well. I can't remember the concentration we had of that one, but you can say measure 10 cubic centimeters of hydrogen peroxide, or of maybe 10% hydrogen peroxide into a beaker. Yeah, and then you can go on and say dip a 5mm filter paper. So this is something that's already been given to you in the question. Okay, so you're pulling out from the question what the answer should be. So you dip a 5mm paper into the extracts that you've collected from the centrifuge, and that's basically like you collecting your catalase. Yeah, and then you drop the paper into the hydrogen peroxide and you measure the time it takes. So always remember to put that, measure the time it takes for the filter paper to rise to the top. Okay, and also write something like record your answer. The other thing is they would like to see you say that repeat each experiment 3 times. The reason is that this shows that you are a good experimental planner. I'm just going to leave this as experiment and just write 3 times. This shows that you're a good experimental planner and you're willing to like do extra work so that you can ensure that your results are accurate. So you can say repeat each experiment 3 times and take the mean value. That would also give you marks. The other thing that students often miss out on, which is an easy 1 mark for questions like this, is that you have to include a control. So you say in another beaker, maybe put just distilled water and put the filter paper inside, and that would basically be like a comparison. So putting a control is always a good idea. Now this is just 6 marks for this question, but I think we've probably come up with like 8 or 9 points because some of them have been like 2 in 1. So if you can write all of these down, you will get full marks. Don't forget, the key things that can easily get you marks when you come to the planning question is taking 5 different samples, repeating the experiment if it's the type of experiment where you can repeat it 3 times and taking a mean, setting up a control. Those are already 3 marks that you will get if you had a question like this. So everything else that is about the experiment, you can pull from the questions that are already in the paper. So don't be afraid of this question at all. And I hope this has been helpful. If it hasn't, leave a comment in the video and I will speak to it again. Something else I should have said on the other video is just that on the other slide rather, is that if you are working with enzymes, another thing that would easily give you marks is to say that you are keeping the temperature constant. You know from chapter 3 that enzymes are very, very sensitive to temperature. So just by adding that, that gives you 4 marks if we add the other 3 that I mentioned earlier. So there is a chance, like if you had an enzymes question, that you could easily get 4 marks out of perhaps 7, and the rest of it would come from you just planning the experiment. So these are easy marks. I often tell students paper 5 is actually the easiest one of all the papers, of the experimental papers. You can easily make mistakes in paper 3 and you have a lot of time pressure in paper 3, but in paper 5 it's just you sitting and acting as if you are preparing someone else to do an experiment. So it should be very easy. All right, let's look at the next question. It says, in a different investigation, the student determined the effect of hydrogen peroxide concentration on the activity of catalase. So if you already understand enzymes, this is basically saying what is the effect of substrate concentration on the catalase, which is the enzyme. So over here they say 5 cubic centimeters of hydrogen peroxide was placed in a test tube at 25 degrees. So note, they've specified temperature. So if you had done that in the previous question, you would also get marks. And look at what they've also done here with like the catalase. So in our case, we said 0.1 gram of carrot extract, which would basically be like the source of our catalase. Here they've sort of specified 0.2 grams of dried yeast was added to the test tube so that the yeast cells formed a suspension in the hydrogen peroxide solution. So the yeast is the one that contains catalase in this case. A gas syringe was quickly connected to the test tube using a bung and delivery tube as shown in figure 1.6. The volume of gas collected after 1 minute was measured. Now, what is the dependent variable in this investigation? Now, again, recall I said the dependent variable is the result of the experiment. I think that's a good way to think about it. It is the thing that happens due to the setup of the experiment. The independent variable is something that you put into the experiment. This is a way to think about it in order to help you clarify it when you're going through questions like this. In this case, the result of the experiment is what the student measured, which is the volume of gas collected after 1 minute. We know that that gas was oxygen. So you can basically come here and say the volume of gas collected, but be very clear that it is the volume collected after 1 minute, because they've specified that. It meant after, like beyond that, they were not calculating, and before that, they were not checking. So after 1 minute is also important, and you will get your full marks there. It's just 1 mark. All right. Okay, moving on. To investigate the effect of hydrogen peroxide concentration on the activity of catalase in yeast, the student first prepared a range of known concentrations of hydrogen peroxide. The student was given 6% stock solution of hydrogen peroxide. The student decided to use the stock solution to make hydrogen peroxide solutions with concentrations of 3.3%, 3.6%, 4.8%, and 5.4%. The student made 50 cubic centimeters of each solution. Complete table 1.1 to show how these solutions could be made by proportional dilution of the 6% stock solution of hydrogen peroxide. Now, this is not scary, okay? This is not, I actually find proportional dilution to be easier to do and also easier for students to grasp, because you simply have to know the formula. Let me grab my pen here. You have to know this formula: C1V1 is equal to C2V2. And if you're a chemistry student, you already know how to get these in. So you already know that V2 is 50 cubic centimeters, right? That's your V2. And you know that C2 would be any of these, because those are the concentrations that you made. And then your C1 is 6%. Okay? And your V1 is what you don't know. You don't know how much of V1 you would add. But C2 is all of these over here. That's C2 all the way down to 5.4. So over here, they've left a thing here. They said volume of, and they've sort of left it blank. This is the volume of water. So they're asking, how much of the stock solution would you take and how much water would you then add? Since you're already adding up to 50 cubic centimeters, you simply need to figure out what your V1 would be over here. Once you figure out V1, then you subtract that from 50, and that would be the amount of water that you need. So let's say, for example, this was, let's see, percentage of concentration of hydrogen peroxide. So let's say C2 was 3. I'm just going to erase these here. I hope I don't have to do any conversions. I've actually just woken up, by the way, so I hope I'm doing all of these right. But I promised you guys to do this video, so here we are. If I get anything wrong, please feel free to write in the comments, but I doubt I will. I don't think I would. Anyway, so C1 is 6. All right. We don't know what V1 is. We know that C2 for this one is 3. And we know that we want to make 50 cubic centimeters of 3. So if we multiply this, that's actually 6V1 is equal to 150. Okay, and then V1 will be equal to 150 divided by 6, which in this case would be 25. All right. And this actually we didn't need to work out because it already just tells you it's half of the concentration, so you can just tell that it's sort of like half the volume of the stock solution and half the volume of the water, half the volume of the final volume rather. Anyway, so over here volume of stock solution will be 25, and then volume of water will then be 25. I hope that makes sense. And you can do the same with 3.6, but you will need a calculator. This will not be that anymore. Let's see. So with 3.6, it would just be, it's my pen. Okay, for some reason my pen is refusing to select. There we go. 3.6 times 50. I've got my calculator close by, so let's see what that would be. That's 180. Okay, so 6V1 is going to be equal to 180. And when you divide that, it's going to give you 30. So that means V1, which would be V for the stock solution, would be 30 cubic centimeters, and your volume of water for you to make it up to 50 will be 20 cubic centimeters. And a good way for you to check if you're doing the right thing is just look at what the concentration of hydrogen peroxide is. As the concentration of hydrogen peroxide is increasing, you need to have a higher volume of stock solution. So it basically, think about it as maybe you're making like honey, like a concentration of honey water. If you want the water to be sweeter, you will need to have more honey, so a higher volume of honey. So think about it that way. So that's one way for you to do a quick check to say, okay, am I doing this right? Also note that this is just 2 marks. So you will need to get, at least for every 2, you probably get 1 mark. So you need to get the pairs correctly for you to get full marks. Bear in mind also that in A-level biology, there is no such thing as half marks. So nobody's going to look at your paper and say, they got this one right, but they didn't get this one right, so I'm going to give them half. That doesn't happen with the examiners. They are not allowed to do that. So always make sure you get your answers as correctly as possible. So I will leave the rest for you to do and move on to the last part of the paper. Okay, this is now the last part of question 1 and coming to the end of the video. It says the student made 2 conclusions. As the concentration of hydrogen peroxide increases, the volume of oxygen produced after 1 minute increases. The Vmax of Catalase is 88 cubic centimeters. Use the volume shown in figure 1.7 to evaluate these conclusions. Now, whenever you see the word evaluate, it means that you should look at whatever you've been given to evaluate objectively. It doesn't mean you must agree with it or you must disagree with it. In fact, you can do both, okay? So always have that in mind whenever you see the word evaluate. Now let's look at what they've told us. As the concentration of hydrogen peroxide increases, the volume of oxygen produced after 1 minute increases. So that seems to be accurate, that indeed when you pair the percentage of hydrogen peroxide with the volume of gas at 3 minutes, let me just find my pen again. So at 3 minutes, I believe this is 3 minutes here, it's sitting at about 36 or thereabout, and obviously as you go on it seems to increase even more. So you can say yes indeed, number 1, yes, as the volume increases, the volume of hydrogen peroxide increases, more gas seems to be collected. However, the second part says the Vmax of Catalase is 88 cubic centimeters. Now, we can't say that this equation or this graph has reached Vmax. Remember your enzymes graph. I'm just going to draw a little one here at the bottom. For you to find Vmax, your graph needs to reach what we call a plateau. So this part of it needs to be flat. That's the second part of it. Then whatever the value here is would be your Vmax. Okay, but we don't have that here. So we can't say that the Vmax is 88 at all, because at 88, it doesn't seem as though the graph has plateaued. The graph seems to be increasing still. So you can go ahead and say it is difficult, or we cannot conclude. Rather, you don't need to say it is difficult because it's not. You can say we can't conclude that Vmax is 88 cubic centimeters because the graph hasn't plateaued. So you write in fuller sentences, don't write like me, okay? I've just woken up, so my brain is still booting and my pen likes to act up. So we can't conclude that Vmax is 88 cubic centimeters because there is no plateau in the graph. So that's another point. And remember, our first point was, yes, indeed, the graph does seem to show. And you can use the words seem to show, because again, you don't know if this is actually like credible results. But the graph seems to show that as the volume of oxygen, as the percentage concentration of hydrogen peroxide increases, the volume of gas collected after 1 minute also seems to increase. But that again seems a bit odd because hydrogen peroxide is the substrate, and for some reason the graph is increasing as though the enzyme is not being fully occupied. If you don't understand what that means, I highly suggest that you watch my videos on enzymes, because then it would make sense. But there's one more point that we need to make. It says the volume of oxygen produced after 1 minute increases. Now, because what we are doing here, I believe, is working with yeast, right? The student was working with yeast cells. yeast cells may not necessarily produce only oxygen, even though they have catalase. Like, yes, they have catalase. Catalase is acting on hydrogen peroxide, but they also contain other enzymes because yeast cells are alive and they have lots of other things going on. They don't simply exist to break down hydrogen peroxide. So the idea that the gas that we're collecting is oxygen is debatable. So you can say, cannot conclude that oxygen is the only gas being collected. Okay, is the only gas being collected. My writing today is so bad. It feels like, it looks like, you know, when a chicken steps in mud and then just steps on paper. And just sort of like, you know how chickens are just like stepping everywhere? That's literally how I think this looks like. But anyway, you can't conclude that oxygen is the only gas that's being collected, and you can write it there because yeasts have other enzymes in them. And that, I promise you, will get you full marks. I'm going to stop here, and the reason why I haven't done the second question is because I don't want these videos to be too long. I thought I would keep this one to 20 minutes, so 25, but once I get into the explanation, I think I just tend to go into a lot of detail, but I hope that it has been helpful. Keep leaving comments on that post that I left on the community page, telling me exactly what you need for the exams, and I will do my best to start putting the videos out. I want you guys to excel in your majoring exams. I get a lot of thank yous from you, and I want to say thank you to everyone who has stayed on this channel, subscribed, who continues to promote it. I really appreciate it. But also just want to let you know that every time there's an exam is, I think, when I'll come out of the woodwork to provide some support. So by all means, use this to prepare for your exams, and also share your results with me when they're out. I am rooting for all of you to get A's and B's. So yes, let's make it happen. And I will see you again in the next video. Goodbye.